Starting today, in the last lecture, we talked about the chromosomes. Let's continue to the transcription translation, and during the start, I'm going to a little bit talk about the chromosomes, okay? So imagine your cells, so imagine the DNA, imagine your cell is a big restaurant kitchen, okay? So DNA is a master cookbook, okay, inside the head chef's office. So that means head chef's office is the nucleus, and inside of the head chef's office, inside there is a DNA. That means the master cookbook, okay? This cookbook, this DNA, I mean the cookbook is a DNA, okay? It holds all the recipes. So those recipes we call them genes, okay? To make all the dishes in the kitchen needs. So you have about 22,000 different recipes. Imagine in your DNA, in that master cookbook, which we are talking about now, which is DNA, inside of this cookbook, there are 22,000 different recipes, 22,000 different genes. Okay, so each one gives instructions to make specific dish, to make specific protein. So DNA is important. It contains the genes, and those genes are going to make us proteins. Okay, this is the analogy compared to the restaurants. Okay, so step one, what's going to happen? The DNA is a long molecule shaped like a twisted label. Okay, here you can see this is the DNA molecule. It's twisted like it's made of four letters. What kind of letters? A, T, C, G. A means adenine, T means thymine, C means cytosine, and G means guanine. These letters pair like the best friends. So adenine is going to pair with what? Adenine is going to pair with thymine, and the guanine is going to pair with cytosine, right? The small segment of the DNA that contains instructions for one protein is called a gene, okay? So this portion of the DNA, this is the portion of the DNA, a small segment of the DNA, small segment of the DNA. This is a small segment. This small segment of the DNA, which contains the instructions for one protein, so from this gene is going to synthesize only one protein, is called gene. Okay, so the DNA is like a library, and each gene is a book. Okay, imagine this DNA is a big library, and this portion of the gene is called what? This is just one book. How many books we have in that library? 22,000 different books. Okay, so imagine a gene like a book and a DNA like a library. So inside of the DNA, you have 22,000 genes, different genes. Okay, DNA is super long. Okay, so it's tightly packed and coiled around the proteins. And we call these proteins what? We call those proteins histones. Okay, we call them histones. So they are super long, they're 2 meters long, they're tightly packed and coiled around the proteins. We call these proteins histones, okay? So histones. And histones and the DNAs, they make up together the nucleus. Okay, they're going to make nucleosomes, and they make a big chromosome then. Okay, so around the proteins, histones, so their DNA is packed around the proteins, which we call histones, and then the whole chromosome. Humans have how many chromosomes? 46 chromosomes in each cell. 23 came from the mom and 23 came from the dad, and each chromosome is like a volume of the recipe book series. So if the DNA is alphabet, if the DNA is alphabet, genes are sentences, okay? And the chromosomes are bookshelves filled with the books, okay? So I will repeat. Imagine the DNA is like an alphabet, okay? Because inside of the DNA, we have the letters G, C, okay? Imagine the DNA is an alphabet, and the genes are like a sentences. These genes are the sentences because they are longer, and the chromosomes, they are like bookshelves, okay? Those chromosomes like bookshelves filled with those books, okay? This is like an analogy. So genes are instructions for making the proteins. We talked a lot, right? Proteins are tools, they're machines, they're builders inside the cell, okay? They do everything. What's the function of the protein? They build the structures. They, for example, they build the muscles, they build the skin, okay? They transport the things, for example, hemoglobin. Hemoglobin is also protein, okay? They carry the oxygen. Also, the proteins, they fight the infections. For example, inside of your body, you have the antibodies which fight the infection. They send messages, for example, insulin. They control all the reaction. For example, insulin is going to control the reaction of the chemicals inside of the body. Okay, so their the flow of information always goes from the DNA to the RNA and then to the protein. This is the flow of information. DNA, RNA, okay, and protein. So from the DNA, this process is called what? This process is called transcription. This is the transcription process. This is what? This is copying the recipe from the book into one note. Imagine this is a big book. Imagine this is a big book, and you're just copying from this book, okay, and one note. Okay, this is one note. Imagine from the book, you're making one note, and this is called the RNA. Okay, from whole book or from the recipe from the book. You have a book full of recipes and you have 20,000 recipes inside to make the food, for example. You have 23,000 recipes. So out of them, copying just one recipe into one node is RNA, and this copying process is called transcription. This node is called messenger RNA. This is called messenger RNA. Okay, so from the RNA, then we're going to synthesize the proteins. Okay, this process is called what? This process is now called translation. This is the translation process. This is like chicken, a kitchen staff, for example, reads the node, that node from the messenger RNA, and cooks the dish. Okay, they're going to make the proteins from that RNA, and this process is called what? Translation. Okay, any questions so far? We talked about the insulin gene in the DNA. Imagine in the part of the DNA, you have the insulin gene, and from the insulin gene, you're going to make the insulin protein. Okay. So, what's the function of that insulin? It lowers your sugar after the meals. Okay. So, you have the hemoglobin gene in the DNA. You're going to make the hemoglobin out of that. The function of the hemoglobin is to carry the oxygen in the red blood cells. You have, for example, the keratin gene. The function of the keratin gene is to make protein, which we call the keratin. And the function of that keratin is to build hair, build nails, and then build your skin. Okay, so also we have the amylase gene. Amylase makes the amylase enzyme. Okay, this is also protein. Its function is to help to digest your carbs in your mouth. Mm, okay, so we have the P53 gene inside of the DNA. So the function of the P53 gene is to make P53 protein. So P53 protein, this protein's function is to stop damaged cells from becoming a cancer cells. Okay, so this is so important. So we have to know everything regarding this. So let's recap. So your body is like a restaurant. I said it before, right? So where the DNA is a master cookbook, okay? And the genes are recipes. And the RNA is a handwritten note passed to the chief, okay, to the chef. And the proteins, they are the final meals that keep the restaurant working smoothly. Restaurant means your body, okay? This is how they are organized. From the DNA, you're going to make the messenger RNA, you're going to copy the genes, and then you're going out of that genes, out of that node, you're going to make the protein, okay? Any questions?
Oh, understandable.
Clear? So central
Yes.
dogma describes how genetic information flows in your cells, okay? For it, it's from the DNA to the RNA and you're making a protein. This means DNA holds the instructions, RNA is a message copy, and the proteins, they are final working product. Out of DNA instructions, you're making the working product itself, okay? So DNA, as I said before, original recipe book, okay? Locked where? It's locked in the safe manager's office or in the chef's office. And the RNA, this is the handwritten note of that recipe taken to the kitchen. And the protein is the cake or cookie baked using that recipe. Okay. So step one, what's going to happen? We are first going to synthesize from that DNA RNA molecule. And this process is called what? Transcription. From DNA to RNA, it's transcription process. Okay. This is like copying one recipe from the DNA into the node. Okay, where this process is happening? This is happening in the nucleus, inside of the nucleus. Okay, good. What's the product of the transcription? It's happening in nucleus. Okay, what's the product of it? Its product is a node. We call it messenger RNA. Okay, its product is a messenger RNA. This is a working copy of the gene. So a messenger RNA, this is single-stranded. It's not double-stranded. Let's look at the RNA. So this is a DNA molecule. As you can see here, this is the RNA molecule. The difference is this is one strand and this is two strand. Okay, it has two strand. So in RNA, you have the thymine, cytosine, guanine, and adenine. But in the messenger RNA, instead of thymine, you have the uracil, adenine, guanine, and cytosine. Okay, messenger RNA uses uracil instead of thymine. Okay, and the sugar is called the ribose sugar, and the sugar in the DNA is called what? Deoxyribose sugar, and the sugar in the DNA is called what? Deoxyribose sugar. Okay, this is the difference. So from the RNA, so as you can see here, from that cookbook, you just note out of the one from one gene, you're going to make one messenger RNA. And this messenger RNA is going to ribosome. Okay, ribosome is a chef. Okay, this is a chef. This chef is reading what? The recipe. It's reading the recipe, which is messenger RNA, and it's cooking what? It's cooking the dish, and those dishes are proteins. It's cooking a dish, we call it proteins. Using what? They're using the ingredients. What are the ingredients? Those ingredients are amino acids. And where is this process is happening? In the cytoplasm. What's the name of this process? This is translation. When the chef cooks a meal, which is protein, using the ingredients, amino acids, in the cytoplasm, is called translation process. And what's the product of this reaction? The product of the transcription is messenger RNA, and the product of this translation is what? This is the protein. Okay, protein chain. Good. Any question? No? We talked about the amylase, we talked about the insulin genes. Imagine this is the insulin gene. From the insulin gene, it's going to go and it's going to synthesize the protein. Which protein? For example, insulin. And the function of this insulin to lower your blood sugar. Okay, so maybe call it collagen gene. This is the collagen gene in the DNA. You're copying a node. Your chef is making you the collagen protein, and this protein is giving your skin strength and structure also. So in our muscles, we have the actin. Okay, in the DNA, we have the actin gene, and this actin gene is going to make us actin protein. And from this actin protein builds muscles and gives the cell its shape. Okay, uh, amylase gene. This amylase gene is going to make us amylase protein. This is enzyme, and it's going to digest the starches. Okay, this is how protein synthesis is important. Okay. Many diseases are caused when the DNA mutations lead to faulty RNA, which makes defective protein. So mutation means you have changes in the sequence of the nucleotides, okay, or missing up the nucleotides, or deletions, inversions, translocations, all of them are DNA mutations. And those mutations lead to what? They lead to faulty RNA. This RNA will be wrong. And if the RNA is wrong, you're making what? You're making the defective proteins. Okay, that's why it's important to know these processes. For example, we have the messenger RNA vaccines. We have the messenger RNA vaccines, for example, COVID-19 vaccine. Okay, they are based on this exact principle. They deliver a safe RNA recipe for your cells to make harmless protein so your immune system can learn to fight it. So you're getting that vaccine, they are injecting to you the messenger RNA, safe messenger RNA, okay? This safe RNA, which is given to your cells, they make what? Protein. They're going to make the protein, antibodies that can learn to fight against it, okay? When you have the real virus, so your body knows how to fight it. This is how the vaccine is going to work. Okay, any questions so far?
No.
Okay, let's continue in the transcription processes. So um, imagine the DNA like a massive library. Okay, so transcription, this is making an RNA copy of the specific part of the DNA, right? This RNA is called what? Messenger RNA. Imagine the DNA, it's like a massive library in your body. You only need one page. I'm saying DNA is massive library. You only need one page. You only need one gene to cook one recipe, right? You don't photocopy the whole book, okay? Just for one page, you're not going to copy the whole book. In order to make one meal, you're not going to copy the whole book. You just make a note of small portion of that book, right? That photocopied page is called messenger RNA. DNA is a big library, okay? So you're going to just, in order to make one protein, in order to make one meal, you're just going to copy just one recipe out of that. You don't just copy all of them. And that messenger RNA is sent out the kitchen, okay, cell's kitchen to the dish, which is protein, okay? So what's happening? So replication and the transcription, you have to know about them. So what's replication? Replication meaning this is the copy of the entire DNA for the cell division. Okay, so replication means this is the copying the whole book. This is the copy of entire DNA. So transcription means this is making a copy of one gene as a messenger RNA to make proteins. Replication is very important for what? It's very important for the cell division, for the mitosis, for the meiosis, and transcription is important for the protein synthesis. You have to know the difference. Replication, copying the whole book, and the transcription is copying just one gene, copying just one page of that book, okay? To make proteins, to make the dish, to make the good dinner, for example, okay? So DNA stays in the nucleus, it's a locked library. RNA is made in the nucleus, right? It's a small copy of that one recipe, and the RNA is going to leave the nucleus to help to build the proteins. Okay. So we have two types of transcription products. So we have the types of the messenger RNA as well. We have two types of the messenger RNA. The first is called polycistronic. The second is called monocistronic. Okay. So polycistronic means okay. This is one. Okay. Polycistronic makes it's like E. coli. Okay. Prokaryote group related genes. Okay. But eukaryotes, all the eukaryotic messenger RNA is going to synthesize only one protein. Okay, for example, human cells, they're going to synthesize only one protein from one messenger RNA. But from one messenger RNA, the prokaryotic messenger RNA is going to synthesize what? Several proteins. Okay, this is called polycystin. Polycystin messenger RNA in prokaryotic cells, there is one long messenger RNA. You see, this is one long messenger RNA which has multiple start lines, start line, start line, start line. You see the start lines? And makes multiple proteins from one message. From one message, they are synthesizing multiple proteins. Imagine one node with three recipes. In one node, you will have three recipes. So cake, pie, and cookies. Several cake, for example, pie, and the cookies. So in one node, you have three recipes. But in monocistronic messenger RNA, which is in eukaryotic cells, for example, in the human cells, one messenger RNA contains only one recipe. It's going to synthesize one protein. Okay, you're going to write only one node for one dish on. Okay, good. In the 5' end, we talked about the 5' during the our topic which we talked about the nucleic acids. Please watch if you haven't watched it. In the 5' in the 5' in eukaryotes, 5' end contains M7G. So this protects the messenger RNA. And in the 3' end, you have the poly A tail, also for protection and for the transport of the messenger RNA. These parts are not translated, okay? Those parts are not translated. They like the packaging, okay? Any questions? So let's recap, let's summarize that you can't forget. So transcription is copying a small part of the DNA into a working copy of messenger RNA so that the cell can use it to make proteins. In prokaryotes, one messenger RNA makes many proteins. This messenger RNA makes many proteins, but in eukaryotes, one messenger RNA is going to make only one protein. It's monocistron. Okay. Any questions regarding this?
No.
Okay, let's shift again. So we have strands, different strands. What's the difference between them? Let's learn the strands of the DNA. We have the template strand. Also, we call the template strand. You know what's template strand? So this is a template strand. This is the coding strand in the DNA. Okay, with the help of helicase, they opened. We talked about it, right? They opened the strand of the DNA now. Okay, so template strand. This is the strand used to make the RNA. This template strand, it's used for making the RNA molecules. RNA is made by reading this strand. This RNA is made by reading this strand. Okay, why? It's called non-coding. So it's also called, this template strand is also called antisense strand or non-coding strand. Why it's called non-coding? Because it's not directly the code we want. Okay, it's not directly the code that we want. It's the reverse blueprint for the real code. Okay, so that's why. So what's coding strand or what's sense strand? Sense strand or what's coding strand? It's not used during the transcription. Okay, this coding strand or sense strand or non-template strand, we call it in three names. Template strand we call non-sense strand, non-coding strand, and template strand. Okay, the question might tell you the coding strand of the DNA blah blah blah, please identify the messenger RNA synthesized from it. So you will be given these kind of terminologies, so you have to know it for the exam purposes as well. So coding strands, they are not used during the transcription. They look exactly like the messenger RNA. These strands look exactly the same to the, for example, the messenger RNA, which was synthesized from the template strand, exactly looks like to the coding strand, okay? Except in the messenger RNA, instead of the thymine, which is inside the DNA, they have the uracil in the messenger RNA, okay? It's the strand we want, but we don't copy from it. We use the template to make its match, okay? To make its match, actually, we need the coding strand, but we cannot directly use this coding strand. We use instead the template strand and we synthesize this kind of messenger RNA, which looks like the coding strand. Okay, good. Any question? So we are making the mirror
No.
image template strand. Okay, you are using the mirror image of the coding strand, which is template strand, to get the right final image, to get the messenger RNA like a coding strand. Okay, so which machine is doing it? Which machine is doing it? We call it RNA polymerase. Okay, it's called RNA polymerase. So this is the RNA polymerase. Let me this is what? This is the RNA polymerase. Okay, so this is the RNA polymerase. So this is the RNA polymerase. So what it does, it's like a copy machine. It's like a printer. It's like a copy machine. Okay, it finds the promoter. We call it promoter recognition site. Okay, promoter where the transcription starts. Okay, promoter recognition site for what? For RNA polymerase. Okay, it finds the start site. We call it promoter. Okay, it binds to the DNA. It opens the strand. It's going to open the strand and build the RNA by matching base pairs with a template strand. Okay, it's going to go in this way. It's going to open. Now I will show you. So these RNA polymerase is going to open the DNA molecule. It's going to go through this way, okay? It adds nucleotides only at the 3' end, meaning it moves along the template in the 3' to 5' direction and makes RNA in the 5' to 3'. So it's moving in the DNA molecule starting from the 3', going to the 5', okay? And synthesizing the new messenger RNA starting from 5'. It's going in this way, okay? The RNA polymerases, they never use the coding strand. It uses only template strand. Where the template strand here? Template strand, never ever forget. It's always starting with 3' and ending in the 5', okay? And synthesized messenger RNA from it, this is the messenger RNA, is always starting from 5' and ending in the 3'. Good? Any questions so far? What's promoter? Here I talked to you about the promoters. So this is the promoter region, right? This is the promoter. So promoter is a special DNA sequence upstream before the gene, okay? Before the genes. It's like start here, sticky note for the RNA polymerase, okay? Tells the enzyme where to land and begin copying, okay? Any questions? What, how the RNA is built, DNA unwinding the region on the gene, RNA polymerase attached to the promoter, and then it starts reading the template strand from the 3' to the 5'. It's building the complementary RNA starting from 5' to 3', okay? If the DNA has adenine, RNA gets uracil. If the DNA has thymine, RNA gets adenine. If the DNA is cytosine, RNA gets guanine. If the DNA is guanine, RNA gets cytosine, okay? Deal. Any questions regarding this? This is the example which is given here. Okay, you have to read it. Any questions?
You said
Regard.
template strand starts with three, end with 5', right? And what which one starts with 5 and ends with 3?
Okay, so this is a DNA molecule, right? This is a DNA molecule. So this DNA molecule is this strand is called what? This strand is coding strand. How I know this is coding strand? Because here it's written 5', okay? Where the RNA polymerase here, this is the RNA polymerase. So here I have 5', that means it's coding strand, okay? Why? Because RNA polymerase always reads from the 3' to the 5'. It's a reading point. Where it's 3', that means this is what? This is the template. This is a template strand. So the template is always from 3' to 5', Okay, but when it moves, when the RNA polymerase move, this is the RNA polymerase, when it moves, what's going to happen? It's starting to release the messenger RNA. So how messenger RNA is released? If the if the template strand starting from the 3 prime, if I release here messenger RNA, this messenger RNA will be synthesized starting with 5 prime, always. Okay, and the end of the messenger RNA will be 3 prime. At the end, you will have this kind of messenger RNA where the start is 5 prime and the finish is 3 prime. Good?
Mm-hmm. Yeah.
Understood? Could I explain you?
Yeah. Yeah.
Hmm, good. So what's going on next? Here, RNA polymerase keeps building the RNA strand until it reaches the special stop signal in the DNA. Okay, in the DNA we have special signal which is called stop signal. Okay, this is called termination sequence. Once it hits that stop signal, once the RNA polymerase hits the stop signal, the RNA polymerase lets go of the DNA and the new RNA strand is made. This process is now done. You have completed the pre-messenger RNA now. Okay, so stop codon is also important. Good. Now it's called the transcript. So the new RNA is complementary to the template strand and almost identical to the coding strand, except the thymine are replaced by uracil. Okay, so what's post-transcriptional processing in eukaryotes? So before the messenger RNA can be used to make the protein, it gets a little makeover to protect and prepare it. Okay, so two things are going to happen. First is a special guanine cap, we call it M7G cap, is added to the front, is added to the 5 prime of the messenger RNA. See, this is the messenger RNA. We are adding what? We are adding the cap, and this cap's name is what? M7G cap. Okay, what its purpose? Why we are adding it? It's protecting the RNA from being degraded inside of the nucleus. It helps it get recognized by the ribosomes. Okay, think of it like putting a helmet on your RNA to protect it. Okay, so what's poly A tail? You see the poly A tail, which is in the 3 prime. This poly A tail, this is the string of adenine bases. It's added to the end, 3 prime end, and this is the poly A tail done by poly A polymerase. Okay, poly A polymerase is going to add that. What its purpose? Its purpose is to protect the RNA. We are protecting RNA with it. It helps with export from the nucleus. Okay, so it helps to export from the nucleus. These poly A tails are exporting this RNA molecule from the nucleus to outside. It's like a ribbon tail on your RNA, so it doesn't fall apart in the cytoplasm. Okay, so it controls how long RNA survives in the cell also. These are the purposes of the poly A tails. Okay, so G5 5 prime cap, so we call it M7G. This is added to the beginning of the messenger RNA. Poly A tail to the end. Poly A tails increase the stability. They aid in the export of this messenger RNA out of the nucleus. The function of the 5 prime cap, it is protect and help ribosome binding also. Okay, they help to the ribosome binding also. Okay, good. Any questions? Let's look closer. So the raw messenger RNA straight from the transcribed is called heterogeneous nuclear RNA. So I'm not going to talk about it very much, but here you can see now the process. So this RNA is like a rough draft. It's not ready to make proteins yet. Okay, this messenger RNA raw. It's not going to make the proteins yet. It needs editing. What kind of editing? What needs to be done? Three main steps. First of all, splicing. What's splicing in the messenger RNA? So this RNA, we call it heterogeneous nuclear RNA because heterogeneous nuclear RNA. This RNA needs modification. Why? Because We should remove the junk introns. So heterogeneous RNA, now heterogeneous nuclear RNA, they contain the exons. Okay, these parts, those parts are called what? They are called the exons, and they are coding regions, important parts. And also we have the introns, and those introns, they are non-coding regions, and they are useless. Those parts are useless. Those parts are useless those introns, and this process is called what? This process is called splicing. And I'm going to join together what? I'm going to join together those exons, those coding regions. I have to join. I have to remove the introns. Okay? Done? Done by the machine called spliceosome. We have a special machine which we call what? Spliceosomes. What? Spliceosomes. Okay? They are complex of the proteins and the RNAs together. Okay? So think of it like of writing an essay and deleting all the extra nonsense, only keeping the good sentences. You're making an essay, you have a lot of junk information inside, so you're going to remove them and you only leave the good sentences. It looks like this. Okay, so what's 5' GTP cap addition? A special guanine GTP is added to which portion? To the, this is GTP cap, to the 5'. Okay, it's added to the 5' end. Its purpose, I said, right? It's the recognition for the ribosomes, and also it protects the RNA from being destroyed. Okay, it's like putting a helmet on the messenger RNA before it leaves the nucleus. So what's poly-A tail adhesion? This is the stability ribbon. Okay, the string of adenines. Can you see here the string of the adenines? We call it poly-A tails. Added to which part of the messenger RNA? To the 3' end. Okay, it protects the messenger RNA from breakdown. It also helps to move the nucleus from the nucleus to the cytoplasm, and also it regulates the lifespan of the messenger RNA. Okay, it's like putting a tag and a ribbon on your luggage before boarding a plane. Okay, have you ever been in the plane?
No.
If you're in the plane, so if you have a luggage, they're going to tag your luggage. Okay, they're going to tag your luggage in order to let your luggage to go to the plane, in order to get the plane. Okay, that's why the poly tail is important for the transformation also. Okay, it helps to move because it's traveling from the nucleus to the cytoplasm, and this poly tail, like a tag, it's like a documentation to leave the nucleus from the nucleus to the cytoplasm. Okay, and that results, and this one is called mature messenger RNA. It's shortened because we removed the introns. It's protected with 5' cap and 3' tail. It's ready to leave the nucleus, and it's ready to be translated into the protein. Okay, any questions?
No.
This is the difference between the DNA replication and RNA transcription. So we talked about, so during the DNA replication, we use the DNA polymerase. During the RNA transcription, we use RNA polymerase. And both of them are occurring in the nucleus, okay? So I think it's not a problem for you. And types and the functions of the RNA. So we have 3 types of the RNA, you know it, right? So first of all
Yeah.
is messenger RNA. So messenger RNA, it carries the instructions from the DNA. It's carrying the instructions, this messenger RNA carrying the instructions from DNA to the ribosomes, right? And also it contains what? 5' cap and the 3' poly tail to protect it during its journey. We have the rRNA. This rRNA, it builds the ribosome, the actual place where the proteins are made, this ribosome, okay? It acts as a scaffold and helps hold everything together. It's going to hold the messenger RNA and tRNA together, okay? It's holding everything. This rRNA is making the ribosome, and the function of this ribosome to hold the messenger RNA and tRNA while making the proteins. And the function of the tRNA, so it brings the amino acids to the ribosomes. It's going to bring the amino acids to the ribosomes. Okay, each transport RNA has an anticodon. It has the anticodon region. We call it anticodon that match to the messenger RNA codon. In the messenger RNA, we have the codons, and the anticodons and codons are going to match, and the amino acids are gonna add together. Right amino acid is added. Okay, it's like delivery person bringing each ingredient to the chef at the exact right moment. Okay, any questions? And all three RNAs are moving to the cytoplasm for translation. So in the translation process, all RNAs are in the cytoplasm. Okay. Any questions so far?
No.
So then the translation begins. So in the translation process, you have the messenger RNA, you have the ribosome, you have tRNA, you have amino acids, and you're making the proteins, right? So what's codon? Codon, it's three nucleotides in the messenger RNA is called codon, okay? Three nucleotides in the messenger RNA, we call them codons, okay? Only three. And each codon is going to make one amino acid, okay? So each codon is responsible to only one amino acid. How many codons we have totally? 64 codons. How I calculated those 64 codons? Imagine I have how many nucleotides? I have adenine, guanine, cytosine, and uracil, right? I have four nucleotides. And out of that four nucleotides, but in the codon, I have three spaces. One, two, three. Using those four nucleotides, I have to, using those four nucleotides in any order, in any order, I have to make codons. And how many possible codons can I make? This is just calculation. 4 in the power of 3. We have 4 nucleotides, and each time in each combination, I have to use 3 of them. How many codons will I have? 4 multiplied to the 4 and also multiplying to 4. At the end, I'm having 64 codons. Okay? But I said one codon is responsible to one amino acid. This is amino acid, one codon for one amino, but I have 64 codons, but I only have 20 amino acids. So multiple codons can code for the same amino acid, okay? Multiple codons, for example, these four codons can be coded for the same amino acid, okay? This is called redundancy, okay? So four codons or three codons or two codons for one of them, okay? Think of codons like three-letter words, okay? So AUG, adenine, uracil, guanine codes for the methionine, uracil for the phenylalanine, guanine, cytosine for the glycine, for example, okay? So we have the start codons and we also have the stop codons. Okay, start codons and the stop codons. So start codon is always adenine, uracil, guanine. This is the start codon, which codes for the methionine amino acid. It codes for methionine amino acid. How it's written? Methionine. Okay, it codes for methionine. Every protein, each protein starts with methionine. And we have the stop codons also. Those stop codons are uracil, adenine, uracil, adenine, guanine. These don't code for any amino acids. They do not code for any amino acid. They just stop the translation process. Okay, start, it's a green light to make the proteins, and stop codon is for the red light. If you see those sequences of amino acids, you should think that they are stop codons, okay? Sometimes in the exam, you might be asked what, or you might be asked to find the stop codons, so you have to memorize them by heart. These are the tasks you can do it yourself, okay? So what else? How the translation process is going to work. So we have anticodons where? Anticodons in the tRNA, and we have the codons in the messenger RNAs, they have to match together. Codons and anticodons, they should match. Anticodon, this is a 3-base sequence on the tRNA. It's in the tRNA. This is tRNA. It pairs with the codon on the messenger RNA. It ensures the right amino acid is added to the chain also, okay? Here the codon CUC is matching to the anticodon to what? Anticodon CUC matching to the codon GAG, okay? tRNA brings the correct amino acid here. Here will be the correct amino acid, okay? Any questions? So as you
No.
can see, this is a translation process. Codon is matching to the anticodons and the correct amino acids are bringing together, okay? So let's talk a little bit about it. So translation where the RNA is used to build the proteins. So this happens in the ribosome. We talked about a lot. So messenger RNA comes out the nucleus. So this messenger RNA coming out just from the nucleus, it's traveling to the cytoplasm and attaching to the ribosome. This is the ribosome. It has a big subunit and it has a small subunits, right? It's made of what? It's made of ribosomal RNAs. So, and the ribosome holds what? Messenger RNA, okay? Holds messenger RNA always. So ribosome is holding messenger RNA like a tape. So it has an A side and it has a P side. Let me show you A side and P side. This is the A side. This is the P side. A side where the new amino acid arrives, okay? So new amino acid arrives. This is the new amino acid where it arrives to the A side. And the P side is where the growing chain peptide is held, okay? Where the peptide is going to made, okay? Peptide bond between the two amino acids. At the end, you can see A side where the new tRNAs are going to come, and as the ribosome moves to this direction, this amino acid will be in the P side of the ribosome, and they are going to be left, these amino acids are going to be left by making one, what? By making one peptide bond, okay? So codons and anticodons should also match together. So this is all we have to talk. We have to talk for today's lecture. This was all. So all the sentences here has been taught during the lectures. So ribosome forms the peptide bond between the amino acids in the P and the A sides. Okay, don't confuse it. So what's making this? What's making this peptide bond? The ribosome. Okay. Then the ribosome slides forward. It's going forward in this direction. It's sliding forward. It's translocating. Okay. So A side where the codon moves into the P side, and a new codon then enters to the A side again. Okay. And this continues codon by codon like beads on a necklace. Okay. So it ends all with stop codons, right? It all ends with stop codons. So S is missing here. So AU, UAA, UAG, and UGA, all of them are stop codons. And when it comes, no tRNA can match it. Okay, no tRNA matches to the stop codon, and in that portion, the ribosome releases the finished protein. It's like saying no more ingredients, recipe complete. Okay, so the final product will be the protein and long chain of the amino acids. We call them polypeptides. Now they have the polypeptides. This is the polypeptide. Okay, and it falls into the functional protein now. Okay. Any questions regarding the codons, anticodons, amino acids, start codons, stop codons, and peptide bonds? Any questions regarding this? Every enzyme, every hormone, and structural component in your body is a protein. Don't forget it, okay? Mutations in the DNA leads to the wrong messenger RNA. This leads to the wrong protein, and that wrong protein leads to the disorders like sickle cell anemia, cystic fibrosis, and so on. And understanding this translation helps you to explain how genetic disorders, antibiotics, even vaccines work. Okay? I hope today's lecture was useful. Any questions to me?
Yeah. No.